Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at the Krebs cycle, which is part of respiration. But let's just remind ourselves about the link reaction. So we know that in the mitochondrial matrix, our pyruvate was decarboxylated, it was oxidized, and coenzyme A came in to pick up our acetyl group. And it was therefore going to transfer the acetyl group to the Krebs cycle. So we have been introduced to our coenzymes before. So NAD, if it is being reduced NAD, has accepted hydrogens. And if reduced NAD is going to release those hydrogens, it's going to become oxidized back to NAD. Well, there's another coenzyme we need to talk about when it comes to the Krebs cycle, and that is FAD. So FAD can become reduced FAD by accepting hydrogens, and it can release those hydrogens by oxidizing back to FAD. So we ended the link reaction in the mitochondrial matrix with this acetyl coenzyme A. So now what's going to happen is that is going to be transferred to the Krebs cycle. So the first thing for us to note with the Krebs cycle, we have oxaloacetate, which is going to be converted into citrate. We then will remove a carbon dioxide, decarboxylation. We will oxidize the molecule. We've got a 5-carbon molecule, which will then go through another decarboxylation and another set of oxidation reactions and substrate level phosphorylation. And that will get us back to oxaloacetate. So let's just take that step at a time. We have acetyl coenzyme A, which comes in and joins the oxaloacetate to the citrate. So we have citrate, which is a 6-carbon molecule, oxaloacetate, which is the 4-carbon molecule, and an acetyl group, which is a 2-carbon molecule. So the acetyl coenzyme A is basically delivering the acetyl group so that it can be combined to the oxaloacetate to form citrate. They are the named molecules that you need to know for the Krebs cycle. Now, the minute the coenzyme A has delivered and transferred the acetyl group, it is now free to return to the link reaction to pick up more acetyl groups. So as we move through them, we've formed the citrate, which is the 6-carbon molecule. So the first thing that needs to happen is this decarboxylation. So the citrate is decarboxylated. We have removed carbon dioxide. Remember, De is the removal, carbox, carbon dioxide. So decarboxylation is the removal of carbon dioxide from this 6-carbon molecule, producing a 5-carbon molecule. But to complete the conversion, we not only remove the carbon dioxide, we also need to remove 2 hydrogens, and therefore dehydrogenation has occurred. So what we've got is this 5-carbon molecule. We have a carbon dioxide molecule, and we have our coenzyme reduced NAD. As we move through from that 5-carbon molecule, what's going to happen is, again, more decarboxylation, removing of carbon dioxide, more dehydrogenation in the fact that we are removing more hydrogens. Now, it is important that you note that we are using 2 coenzymes this time. So we have 2 reduced NADs, and we also have a reduced FAD. So 3 coenzymes that are going to remove from that 5-carbon to get us back to the oxaloacetate. But also what we have to note is that substrate level phosphorylation also occurs. Now, substrate level phosphorylation is the transfer of a phosphate from an intermediate molecule to produce ATP. So ATP is produced, and that is because we have this transfer of a phosphate from the molecule to the ADP to create ATP. So it's important that we note that substrate level phosphorylation is occurring, and through that process of the decarboxylation, the dehydrogenation, and substrate level phosphorylation, we have regenerated oxaloacetate. So we've got back to the original starting point of our 4-carbon molecule. And that can then go around again when more coenzyme A come in with their acetyl group attached. Now, what they love to do in the exam sometimes is they love to give you this cycle, but show you many of the intermediate stages that could be in between. Now, you're only expected to know oxaloacetate and citrate and the acetyl group. However, it is important that you note, if they're giving you a different molecule, look at how many carbons it has. Therefore, if it needs to be converted into an intermediate that is able to go through the cycle, how many carbons would need to be removed? How many decarboxylations would need to occur to remove the carbon dioxide, therefore getting us to that 5-carbon or that 4-carbon. The other point to note is how many hydrogens would need to be removed. So when they do throw a question at you, which is looking at the Krebs cycle, they like to give it as a application, not just memorizing. So do make sure you understand what is happening, how many decarboxylations, how many dehydrogenations, and what are your end products that you get. So if we were to look at the products that were made through the 3 parts of our process of aerobic respiration, in glycolysis we have 2 reduced NADs and we have 2 net ATP molecules. Through the link reaction, because we had 1 glucose molecule converted into 2 pyruvate molecules, as those 2 pyruvate molecules are converted into the acetyl group, we end up with 2 reduced NAD molecules. And from the Krebs cycle, what we are getting is, through that process of the oxaloacetate being converted into citrate and the citrate then being regenerated back into the oxaloacetate, we end up with 6 reduced NADs because we had 2 acetyl groups from 1 glucose molecule. We have 2 reduced FADs and we end up with 2 ATP molecules. So it's really important before we move on to the final stage of aerobic respiration that we know what our end products are. So I hope you have liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.